Now, this might be an odd talk, because I'm actually a philosopher of biology. But I'm still going to talk about experimental evolution. Now, like many others, I will talk about Pseudomonas florensis today. The title is Evolution of Multicellularity, Cheating Done Right. Now, this is a sexy title, but what do I mean here? Well, I'm going to talk about a paper I recently published in Biology and Philosophy with the same title. And I'm talking about ideas mainly coming out of a research program by Paul Rainey, it's been named in the last talks as well. Now, he has introduced recently this idea of ecological scaffolding, which is supposedly able to explain how we can move from single-cell organisms to multicellular organisms. How do we move from one level of Darwinian individuality, from one unit of selection to another? Now, this is Paul Rainey, and together with Katrin Hammerschmidt, Rose, and Kerr, they've published this paper, Life Cycles, Fitness Decoupling, and the Evolution of Multicellularity. Now, fortunately, I don't have to explain this, given the previous talks. But here, what we have is that we have some kind of proto-group emerging. But of course, because cheats arise, cheating types that don't produce this glue, the mats become heavier, they become destabilized, and the mat, in the end, breaks down. It's the doom of this group organism, and we don't have a new life cycle. So how can we have a new Darwinian individual emerging? Now, they've introduced this interesting hypothesis to see cheats actually not as a problem, but as a solution. So I'm here defending these two philosophical ideas, that first of all, we need a conceptual shift from seeing cheats as a problem that needs to be solved for multicellularity to evolve, to the very key for the evolution of multicellularity. Now, first I'm going to sketch the problem, then the solution, and talk a bit about experiment. And much of this is based on work by my supervisor, Samir Okasha. And in his book, Evolution and the Levels of Selection, he wrote, well, multi-celled organisms did not come from nowhere. And a complete evolutionary theory must surely try to explain how they evolved, rather than just taking their existence for granted. So levels of selection other than that of the individual organism must have existed in the past, whether or not they still operate today. Now, traditional explanatory tools by evolutionary biologists, however, already take this level for granted. So it is still a persisting problem in biology to explain how these new levels, the major transitions, actually eventuate. Now, what is the problem? Well, we need to explain how the transition occurred from single-cell organisms to multicellular organisms. The traditional answer here has been cooperation. Much of the work by Michaud has been based on this. Now, Paul Rainey points out that cooperation is not sufficient, however. Right? Imagine a group of single cells perfectly being able to cooperate with each other. Why would they even need to give up their individuality to become part of a bigger whole? Now, they argue that we need two further things here. First of all, a mechanism of group reproduction, and secondly, a mechanism to minimize the adverse effects of cheats. And these need to happen at roughly the same time. The solution here I'm going to sketch via two tools: multi-level selection models and work by philosophers, and secondly, experimental work. Now, Peter Godfrey Smith, in his book Darwinian Populations and Natural Selection, he's my second supervisor, he has provided us with this elegant tool, these Darwinian spaces, in which we can think about Darwinian individuality. Now, we often take humans, us, as this paradigm case for Darwinian individuality. But of course, once cancer arises, this individuality breaks down. Now, the conditions for individuality he sketches here is, first of all, a bottleneck, reproductive specialization between germ and soma cells, and oval integration. It's the division of labor and maintenance of a boundary. And if you think of a buffalo herd, of course, none of these are instantiated. But if you want to move from one level of selection to another, if you want to have new individuals on a higher level emerging, we need slowly, gradually move towards these three properties. Right. Now, how can we do this? Well, Darmuth and Heisler have provided us with this elegant distinction between multi-level selection 1 and multi-level selection 2. And this, I hope, puts to rest many of these disputes in the group selection debate. Now, they have distinguished multi-level selection 1. Here, group selection refers to the effects of group membership on individual fitness. And fitness here is a property of individuals. And from multi-level selection 2, which is here, group selection refers to the change in frequencies of different kinds of groups. Fitness is here properties of groups. Now, when Maynard Smith criticized group selection models, he had something like multi-level selection 2 in mind, that this is actually not group selection, right? Now, what needs to happen, however, if we want to move from one level of selection to another, is that we need to decouple fitness, an idea by Michaud. So we need to move from multi-level selection 1 to multi-level selection 2. But how can this happen? And Paul Rainey here introduces this idea that cheats could serve as a sort of proto-germline probacule that is able to detach itself. Why? Because it does not produce this cell-cell glue, so it is able to detach itself. It's not tied to the fate of the group organism. So we can have some sort of primitive life cycle here emerging that switches between these Winkley spreader mats and mutant provacules. So the mutant provacules, however, once detached, need to switch back to cooperation, so cell-cell glue production, forming a new individual in the end. And there is experimental support for this. And if this happens, we can meaningfully speak of these cells within the mat as working for the organism. And this is then this move from MLS1 to MLS2, fitness decoupling. Okay. Now, what do we get here? Well, we get a slow transition towards a new Darwinian individual. We have a bottleneck during which a probacule marks the beginning of a new life cycle. We have a germline that's being specialized for reproduction of the collective. And there's overall integration of the individuals, forming a new individual rather than just a group. And this, of course, is just marginal. But this is precisely what we need. All right. And then we could put these Winkley spreader mats roughly here, and hopefully further experimental work is able to push them further. Now, let me talk a bit about this experiment. So we have these spatially structured undisturbed microcosms with an ancestral smooth genotype and these mutant Winkley spreader types who quickly take over the surface. They are able to reap the benefits of oxygen. Normally, their fitness would be lower. But importantly, this only works given these appropriate ecological conditions, which is why he introduces the idea of ecological scaffolding. Now, Paul Rainey argues that in order for natural selection to operate, we need certain conditions to apply. Now, some of these conditions need to gradually emerge, but sometimes perhaps the environment can give us these conditions, right? And in some trivial sense, ecological scaffolding always happens because every organism needs the environment to reproduce itself. Now, of course, we have here a tragedy of the commons, spread of mutants that leads to the doom of all, and this is something we need to avoid somehow. Now, the result of this experiment was that conflict between these cooperating Winkley spreader cells and cheats gave evolution something to act upon. These Winkley spreader mats are like Soma, an evolutionary dead end. But what was possible in the cheat embracing regime was that the fitness of mats became actually decoupled from the fitness of the single cells. Now, what are these two regimes? Well, in one of them, we started the life cycle with a cheating cell, and in one of them, we started the life cycle with a cooperating cell. Now, if you think of the work of Michaud, it would seem that this is actually what we want. Cheats need to be purged. But interestingly, the experiment showed that here we had actually some kind of de-Darwinization happening, an idea by Peter Godfrey Smith. So the individual cells within the mat lose their individuality. They become part of this bigger whole. All right. Now, all right, do I have enough time? Initially here, they only tested us with 120 of these spatially undisturbed microcosms, and every microcosm starts with this yellow Winkley spreader genotype, and then we have this setup, the single cell bottleneck, and yeah, I think we have enough time. In phase 1, each of these 120 microcosms is left alone for 6 days, after which the cells are harvested and screened for SM colonies. If no Winkley spreader mat is formed, no SM are found, the line ends, that it goes extinct. If no Winkley spreader mat is present, the multicellular body has not remained intact, while failure to produce M cells stands for a failure to produce propagules. So we have something analogous almost to a sort of group-level individual happening here. Now, in phase 2, which only lasted 3 days, these microcosms continued solely with the newly formed SM cells. They failed to transition back to Winkley spreader cells within this time period. The line was also taken to be extinct. Now, this in the end allowed for the possibility of life cycle enhancing mutations, which are beneficial over this longer time scale of the life cycle, which is important here. Able to outrun life cycle disrupting mutations. All right, so what have we learned from this? Well, Samir Karsha in his book finished his last section on the major transitions with the following conclusion in 2006. The study of evolutionary transitions is still in its infancy, with much empirical work remaining to be done. So it is difficult to say whether the foregoing analysis will prove satisfactory in all respects. But whatever future developments in the field look like, it is likely that multi-level selection will remain crucial for theorizing about evolutionary transition. So what is the conclusion? The cheat-embracing regime decouples the fitness of the mat from the fitness of the cells. The mats with the highest fitness consist of cells with the lower-level individual fitness. Cells start to work for the mat organism. The hypothesis was that cheats and conflict drive the evolution of multicellularity, and we've gained empirical support for this. So the problem of cooperation, group reduction, minimization of adversive cheats can be solved by seeing cheats as the first single-cell propagule, marking the beginning of new life cycle through a bottleneck. This reduces this distinction, and we have some sort of marginal developmental control. So the verdict is, we need a conceptual shift, and this allows us to see the importance of multi-level selection and the importance of ecology. Thank you.
Thank you. Now questions, if you have any. Can I ask something? Oh, okay.
So if the cheats have a different genotype than the cells on the mat, are they really equivalent to a germline, or?
So what needs to happen is, and this is of course only marginal, here we're talking only about the phenotype, and this could happen in several ways, right? So, for example, the cheat might mutate again and then switch back. But what happened in this experiment was that these cells were able to switch back by themselves.